Hello everyone, that's Dr. Shivangi at your disposal. So let's start with the first chapter, that is cell biology or the fundamental units of life. These would be my contents, starting with what is cell biology. So it is a branch of biological science that focuses on the study of cells, which are the fundamental building blocks of life. Every living organism, from the smallest bacteria to the largest animals and plants, everything is composed of cells. So these microscopic structures, they perform all the essential life process, which includes metabolism, growth, reproduction, and response to stimuli. The other thing is that if anything is acellular, will we call that as living? And do we have any cellular acellular organism? Answer is yes. And that a cellular organism, I can say it is a virus, it can be a prion, it can be a viroid. So all these things, they are merely the aggregates of the biomolecules, but they do not form a complete cell. And that is why we categorize them as a borderline between living and non-living. So the bare minimum structure that an organism needs to survive is a cell. And a cell, we can say, is the exact replica of the entire body. So whatever process in general is happening on the macroscopic level is also happening within the cell, be it excretion, be it digestion, or the metabolism, or the cell growth, reproduction, response to stimuli, all of that. Right. So moving on to the next slide. Study of cells helps scientists understand how living organisms function at the most basic level, and that would eventually lead to medicine, advancements in medicine, genetics, and biotechnology. So there are multiple branches which are working at a cellular level, where we see a disease not just in terms of the macroscopic view of it, but we also see it that how is it behaving at the microscopic level. And we are identifying the areas where we can make the amendments and provide the right treatment. So that is how we can, by understanding the correct structural level or the basic level of the cell, will be able to advance in terms of medicine, genetic, and biotechnology. So cell biology, also known as cytology, is the scientific discipline focused on the study of cells, the structure of the cell, the function, the behavior encompassing their composition, organization, and interaction with others. Very basic example that I might take over here is that of the cancerous cells. So because we know the behavior of a normal cell, that is how we are able to determine the abnormal behavior of cancerous cells. Had it not been the structure of cell, we would not have identified the disease as cancer. So basically, whenever we understand a disease, we try to understand it at the grassroots level, which is definitely a cellular composition. We know by definition, it is the fundamental unit of life. Fundamental, that is the most basic. It forms the foundation. It is laid one over the other to give rise to the tissue and then to the organ system and the entire body. Then is this the structural unit and functional unit, because one single cell replicates the functions of the entire human body. One single cell would replicate the functions in the entire human body. So that is why it is the functional unit also. Cells can exist independently as unicellular organism. दो kingdom है unicellular organisms के prokaryotes में. एक kingdom आ जाता है monera, which includes all the bacteria. The second kingdom is the eukaryotes, not all the eukaryotes, obviously. The very first kingdom that incorporates the eukaryotes, which is the kingdom Protista. So Protista contains amoeba, protozoans, euglena, and dinoflagellates, diatoms, etc. And then they can form a part of larger organism, which we see in humans, plants, animals, and fungi. Cells are different in shape, size, and function. And it is very surprising to know that all the cells have different shape so that they can perform different functions. Sorry. For example, we have a neuron, which has a branched shape like this. Then we have an ovum, which is an ovoid cell. Then we have an RBC, which is a biconcave shape. So different cells, different function, different shapes. So if we say that a particular function is governed by the shape of it, obviously yes. Because if you imagine a neuron being a circular, it would not be able to transmit the electrical impulse. But this has to be like wires and in the branching pattern so that the impulses can get collected and can be traversed through it. They share common characteristics such as plasma membrane, cytoplasm, and genetic material. All these things are very common. Some organisms, like bacteria, consist of single cell, while others, like humans, have trillions of specialized cells working together to sustain life. So these are the different cells that we'll encounter in the chapter ahead. We have the squamous epithelium from the mouth, from the oral cavity, and they are very flattened type of cells and they allow for the exchange of substances. The topmost layer, though the stratified squamous type of epithelium is present in the mouth. Then there is ciliated epithelium from the trachea, the cilia. Cilia helps in the movement of mucus. Movement of mucus. Also, it is present in the oviduct. It's present in the oviduct and helps in the movement of ova. Movement of ova. Then columnar cells from the stomach. So muscle fibers, these are the fibers from the heart. And why are they branched? Why are they branched? If you know the answer, please comment down the answer in the comment section below. Then there are muscle fibers from the intestines, the striped muscle fibers. Where do we see it? Striped muscle fibers. We have a specific name for it. What that name is, what this type of muscle is, just identify and let me know down. Okay, this is ovum as discussed, sperm the smallest cell of the body, ovum again the largest cell of the body, neuron is the longest cell of the body, and so on. Cartilage cells are called as chondrocytes, chondrocytes, bone cells are called as osteocytes, so on. Starting with the history of cell, which most of you would like to know and is there in the syllabus for everyone. I think maybe it's the most basic thing as well as and simultaneously the most. Okay, so starting with Robert Hooke. Robert Hooke was the first scientist to observe the cells for the first time. He was the first to observe and describe the cells, and he used a compound microscope. Why is the microscope called as compound? Compound because it uses more than one lens. It uses two lenses at times. That is why we call it as compound microscope. What two lenses is it using? It is using ocular piece, eyepiece, ocular, or the other lens is the objective lens. Right? The two lenses are there, and that is why it is called as a compound microscope. He examined a thin slice of cork. Cork is the outermost part, outermost epidermal layer of the woody plants. Outermost epidermal layer of woody plant, the bark wala jo part hota hai, that is, so that structure is generally dead. So he was the one who noticed the dead cell for the first time. If they ask you who noticed the dead cell for the first time, you would answer Robert Hooke. But he, अगर living cell पूछा तो he will not write Robert Hooke. However, Hooke only observed dead cells. And जब dead cell observe किया तो क्या उसने nucleus observe किया? No, he didn't observe the nucleus. He observed only the cell wall, and that is why the honeycomb-like structure, the honeycomb-like structure, you can see it over here. The honeycomb-like structure. This was the primitive microscope that the Robert Hooke had used. Primitive microscope. And then there you can see the book written by Robert Hooke, book written by Micrographia, Micrography. He's Robert Hooke himself, by Robert Hooke, right? And here we can clearly see the honeycomb, like only because of the cell walls. We do not see any cytoplasm. We do not see any nucleus over here. Honeycomb-like structure. Fine. Moving on to the next scientist, Antony von Leeuwenhoek. He was responsible for the discovery of living cells, and he used the improved microscope lenses and became the first one to observe the living cells. He collected pond water and he saw tiny moving organisms, and which he called as animalcules. Animalcules come is basically the term we use for the locomotory organisms, now known as bacteria and protozoa. They are referred to as animalcules because they are the motile organisms. So obviously they are not going to be the plants, they are going to be animal related, and that is why, because tiny animalcules. He also observed the sperm cells, the RBCs, and muscle fibers. So this is one of the questions that can be asked. Okay, let me highlight the key points from here. He was the first to observe the living cell. First to observe the living cell. 1 point. Second point, he called them as animalcules. He observed bacteria. Third thing, he observed protozoa. Then he observed sperm cells. He observed RBC and muscle cell. So these questions are generally asked in the exam. Moving on to the next scientist. Before that, let's see. It is the picture of Antony von Leeuwenhoek, and this is the microscope that he used. The foundation of cell theory. It was put forward by Matthias Schleiden. He observed the plant cells, so definitely he is going to be a botanist. German botanist. He studied the plant tissues under a microscope and he concluded that all plants are composed of cells. He also proposed that cells are the basic building blocks of plants. Cells are the basic building blocks of plants. Schwann produced he studied it on the animal cells. He was a zoologist and he made the similar observation on the animal cells. He also discovered that animal tissues are also composed of cells. So together they formulated the theory that all living organisms are composed of one or more cells, be it plant or animal, and therefore the cell is the basic unit of structure and function in living thing. Moving on. Then it was Rudolf Virchow. Rudolf Virchow, he gave the principle of cell division. He gave the final principle. He added the, or we also say that he modified the cell theory. He modified the cell theory. He gave the principle omnicellula e cellula, omnicellula e cellula. That is, all cells arise from the pre-existing cells. All cells arise from the pre-existing cells. This is very important. Rudolf Virchow. And this also disapproved the earlier belief of spontaneous generation, the idea that life could arise from non-living matter. So these are the three main postulates. Living organisms composed of one or more cells. Cell is the fundamental unit of life, and all cells arise from pre-existing cells. But that is not an end to the list. There is some advances also. These theories are the newer theories, which we should have a look at. Louis Pasteur, he disapproved the spontaneous generation theory. He was a French scientist, Louis Pasteur. He conducted experiment with swan neck flask. So this flask used to look like this, somewhat like a swan's neck. It was a flask like this. And he proved that microorganisms do not arise spontaneously. This we'll study in evolution again, but come from the pre-existing cells. This supported the virtuous cell division principle. Then, who discovered the Golgi apparatus? It was Camillo Golgi, Italian scientist. He developed the silver staining technique. Very important, silver staining technique, because Golgi apparatus is not visible like that. He discovered that Golgi apparatus, an important organelle involved in modifying and packaging proteins. Moving on, there was a scientist named as Cajal, Santiago Cajal, neuron theory. He was a Spanish neuroscientist and he studied the structure of the nervous system. He provided evidence that neurons are individual cells and not a continuous network, forming the basis of modern neuroscience. Then there was the discovery of electron microscope that enables us to see the ultrastructure. It was invented by Ruska and Knoll. So you don't need to remember Ernst Ruska and Max Knoll, just Ruska and Knoll would do. Sometimes they ask you this question. Not very important though. This allowed scientists to see subcellular structures like mitochondria, ribosome, and lysosomes. Okay, so it led to the discovery of ultrastructure within cells. The structure which we are not able to see with the normal microscope are called as ultrastructure. See, only with electron microscope. Now, why is electron microscope better than light microscope? This is again a question. Please answer in the chat box if you know. Why is electron microscope better than light microscope? I'll be revealing the answer in my next lecture. Before that, if you find the lecture and just try to write it down in the comment section below. Is electron microscope better than the light microscope? DNA as the genetic material, Avery demonstrated that DNA is the hereditary material and not the proteins. We'll read about this in biotech. James Watson and Crick, they gave the double helical structure of the DNA and with the help of the Rosalind Franklin's X-ray diffraction images. So it is a double helical structure of DNA and that revolutionized the genetics and the molecular biology. Fluid mosaic model again. Fluid mosaic model was given by Singer and Nicholson, and this suggests that the plasma membrane is a flexible bilayer of lipids. It swims like this. It is not completely static. It is fluid. That also explains the flexibility of the plasma membrane, which is responsible for causing endocytosis within an amoeba. Right? So that happens. Why? Because the plasma membrane is flexible. And what explains its flexibility is the fluid mosaic model, which suggests that plasma membrane is a flexible bilayer of lipids and there are proteins embedded in it like this. Right? There is some intrinsic proteins, some peripheral proteins, and transmembrane proteins. So these are the two types of cells, the prokaryotes and eukaryotes. In prokaryotes, what are the main differences that you need to remember? In prokaryotes, they do not have a nucleus. Instead, they have a nucleoid also called as genophore. Also called as genophore. We can also refer the word fibrillar or incipient nucleus for the same. Fibrillar nucleus or incipient nucleus for the same. Okay, so these are the very old type of nucleus. They do not have a proper nuclear membrane around it. They lack the completely membrane bound organelles. They may have a cell wall. they have capsule at times, and such bacteria or such organisms are more virulent. They may have flagellum, right? They have ribosomes, but their ribosome is 70S type of low molecular weight. In eukaryotic cell, you will find a nucleus, you will find nuclear membrane, which is double membrane bound. Then there are cytoplasm, mitochondria, Golgi. Like there is n number of organelles that is present, endoplasmic reticulum, chloroplast. All of these are the membrane bound organelles. Right. Starting with plasma membrane. Plasma membrane, also known as the cell membrane, is a dynamic, selectively permeable membrane that encloses the cytoplasm of a cell, and it plays a crucial role in maintaining cell integrity, transport of substance, signal transduction, intercellular communication, and so on. When I say intercellular communication, it would mean that plasma membrane is connecting the two cells like this. It the model that describes the plasma membrane is the fluid mosaic model, which suggests that it has a lipid bilayer like this, right? It has a lipid bilayer like this, and it has some proteins embedded. It has some proteins embedded. So basically, it suggests that the lipids are in a wavy motion, or it's in constant motion, that gives it a semi-fluid appearance. It comprises of phospholipids arranged in a bilayer, and they are amphipathic molecules. Why amphipathic? Because they have two types of polarities. They have hydrophilic heads also, and they have hydrophobic tails also. So heads, because they are hydrophilic, that is water loving, they will face the extracellular fluid. They will face the extracellular fluid. For example, this is the cell. If I'm zooming this portion, I'll see that this is made up of lipid bilayer, and this head faces towards the extracellular fluid, and this tail faces inwards. So this is obviously hydrophobic, that is, it has the fear of water, it has the fear of fluid, so it is oriented inwards away from water. Membrane proteins, there are three types of protein. Let me make the structure again. That is how the lipid bilayer look like. So if it is only passing or is halfway, it is integral protein. If it is completely crossing the membrane, then it is transmembrane protein. And if it is present on the periphery, it is present on the periphery like this. This is the peripheral protein. So we have some extrinsic proteins. This is an extrinsic protein, and these are intrinsic proteins. They all have different functions. Glycoproteins, okay, some of them have sugar molecules or glycogen molecule attached to it. They are referred to as glycoproteins. So two components can be there. If glycogen is attached to the proteins, it is glycoprotein. If it is attached to the bilayer, it is glycolipid. Form the glycocalyx, which function in cell recognition. This is very important. Glycocalyx, function of glycocalyx. It helps in cell recognition, cell adhesion, and protection. Cell recognition, adhesion, and protection. So that is how it looks like. We can see different types of proteins, integral protein partially passing through it. Then there is glycolipid present on the lipid layer, glycoproteins present on the protein layers, carbohydrates, and the surface protein, which is present at the periphery, phospholipids, and so on. Cholesterol can also be seen. Functions of plasma membrane I shall be discussing in the next class. So this was part one of cells at my end. See you next time. Bye bye. Good night.